Hello students, in the last class we have discussed about chromosomes, structure of chromatin fiber, the components of chromatin fibers, which is 40% DNA and 60% histone proteins. Then we have already discussed about the scientists who have discovered chromosomes, why the name chromosome is given to the nuclear substances and the chromatin fiber. Today we will discuss these following topics: structure of chromosome, genes, cell cycle, and what is the need for cell division or formation of new cells. So this first topic, structure of chromosome. Chromosome is made up, as you all know, that chromosomes are made up of chromatin material, and chromatin is a thin thread-like substance. These thin thread-like substances, they are wrapped around histone proteins and they form an octamer. Octamer of 8 histone proteins, it forms nucleosome. So this nucleosome, if we are writing the components, then chromatin fiber wrapped around histone proteins to form an octamer. Octamer, that is a group of 8, and this 8 forms nucleosome. These structures, they are coiled to form the chromatin fiber, chromatin material. Like this, they are wrapped around, and this, it is a single or the monovalent chromatin fiber. When two chromatids or two sister chromatids, they join together at a point, and the point of contact is known as centromere. The point of contact where the two sister chromatids join together, that is known as centromere. The next thing is, this is the structure of, again, chromatin fibers wrapped around histone proteins. All these together form a group of 8, that is the octamer. Then they are called, when they form group of 8, then they are called nucleosomes. These nucleosomes wrapped around to form chromatids. When they join together, the point of contact or the point of attachment is known as centromere. The second topic for today is genes. What is gene? Before this, in the previous lecture, I have explained about the nitrogenous bases. Adenine, guanine, cytosine and thymine. In this, we all know that adenine always pairs with T, guanine always pairs with cytosine, that is with a triple hydrogen bond, A with T by double hydrogen bond, G with C by triple hydrogen bond. They are present on the backbone of DNA to form they are formed present as a ladder. And within these two strands of DNA are complementary to each other. This sequence, by knowing the gene sequence, by knowing the sequence of nitrogenous bases, we can easily find out the sequence of the complementary strand. Nitrogenous base plus sugar plus phosphate forms nucleotide, and the sequence of nucleotide is known as gene, which is the genetic material or the hereditary unit. Genetic material or the hereditary unit, the characters which are transmitted from parents to offspring are transmitted through these genes. Unit. Next topic is cell cycle. Next topic we'll discuss about cell cycle. Cell cycle, it is the series of sequences or series or the steps involved in the life cycle of a cell for the growth, for the formation of new cells or the cell division. Sequence or the series of events. Series of events is known as cell cycle, and it involves two basic series. That is, one is the non-dividing event, which is also known as interphase or the resting phase In this phase, the cell prepares itself for the further division. The name resting phase is a misnomer, as the maximum work like formation or duplication of DNA material, formation of proteins, increase in the cytoplasm, all these things takes place in the interphase stage only. But the name is resting phase since the cell division is not taking place. Second step is the dividing phase. Dividing phase, which involves two type of divisions, mitotic as well as meiotic division. Mitosis takes place in the somatic cells or the vegetative cells. While this meiotic division takes place in the reproductive cells, help in the formation of sperm and egg, or male and the female structures in vegetative parts. This one in vegetative plants, vegetative structures, the vegetative plants, you can say. Now next topic is need for new cell. Need for new cells. Need for new cells. The formula for this is GRR. G is for growth, R is for replacement or repair, 1R is for replacement, and the next is for reproduction. Growth phase, the growth of a cell, like this is the space or the size of an individual, and it is having single cell. It divides into two. Further, this results in the increase in the size of the organism. So this for the growth of an organism, division is required, and the division is mitotic division for the growth purpose. Then repair, the replacement of worn or the wear and tear process continues in the life cycle of all the organisms. So to replace or to remove, to repair the cells or the damaged cells, This process is required. The formation of new cells or cell division is required. Replacement of the dead cell tissues also. Division is required of formation of new cells. The dead cells are replaced by the new cells. Then the reproduction, formation of sperm, egg, ova. All these things requires the replacement formation of new cells. This is all for today. Today's homework. 2 questions I am giving you for today's topic. First, write the structure of chromosome. Structure of chromosome. Describing nucleotides. Second one is what do you mean by genes? What do you mean by genes? That's all for today. Now you complete your homework and do revision of these topics.